Thank you for the kind introduction, Sarah. It's a pleasure for me to show you today how we have integrated the new Genexus next generation sequencing instrument in our daily routine. So before we go into actually operating the instrument, I would like to first show you a little bit the software and how it is used to set up a run on the Genexus sequencer. So the server running this software is actually integrated into the instrument, meaning I can place this instrument anywhere I want and I don't need to have a separate server room. Wherever the instrument is, I have the full package. I have both the NGS instrument as well as also the server running the software. So let's log into the software. So once we log into the software, we are greeted with a dashboard. In this dashboard, we can see previous runs done. We can see also the QC of the samples from the very last run. And it gives us an overview of what has been recently done. All right, so let's start by registering a sample. This is done through samples and create sample. So here we have the option to either register single samples, or if we have batches of samples, we can actually create an Excel file and upload this directly, so we don't have to create one by one each sample. Nevertheless, I will show you now an example of registering a single sample. So first we give it a sample name. I will call it test sample 1. We have the option to tag the sample with a specific project name, for example. And then we choose the application category. In our case, since we use the instrument mainly for the molecular profiling of solid tumors, we would choose solid tumor. Next, we choose the sample type. Here, if you're using the Genexus purification system along with the sequencer, you can choose whether you have an FFP sample, a fresh frozen, a blood sample if you're doing liquid biopsy. In our case now, let's assume we start from already extracted nucleic acid. So let's assume we have a sample where we want to do both DNA and RNA analysis. So we would choose this, DNA and RNA. Next is the cancer type. Let's assume we have a non-small cell lung cancer sample. Next is cancer stage. If it is known, if it is unknown, let's assume in this case we don't know the cancer stage, could be an external sample. Then the percentage of necrosis present in the sample, as well as the percentage of tumor cells. Now the percentage of tumor cells is not a mandatory field, but we highly recommend to enter this because it may affect the accuracy of copy number variation calling. So let's assume this sample has 50% tumor cells. Last but not least, we can choose the gender. In this case, let's choose male, and we can add the collection date of the sample if it is known. Once this is done, we would save the sample, and it is now added to the list of samples, as you can see here on top. So now we have registered our sample in the system. So the next step would be to plan a run for sequencing. And this is done via the Runs tab on top. In this case, we will start from nucleic acid. As I mentioned before, we also have the option to start from samples, for example, from an FFP sample, from blood, in case we would be using the Genexus purification system. So today I will show you how to start from nucleic acid. So the first part would be to give the run a name. In this case it would be, let's call it test plan 1. We have actually the option to mark a field here to automatically generate a report when sequencing and variant calling is done. So we would press next. The next step would be to choose the assay that we want to run. We can run many different assays. Basically any custom AmpliSeq or AmpliSeq HD assay can be done on the instrument, along with also the commercially available assays. In our case here in Basel, for our routine use, we use mainly the Oncomine Precision assay. This you can see here, and also for this assay you actually have different options. So you have the option to run only the DNA part of the assay, or you can run only the RNA part for gene fusion detection, or you can decide to run both DNA and RNA. In this case, let's choose DNA and RNA. The next step is to choose the samples that we want to run with this assay. So here again, now we see the list of samples that we have previously added and that is registered in our system. Let's just choose some random samples. Here, for example, we have one DNA and RNA. Here's another one, and maybe here 2 more. We can click Assign, and now these samples have been assigned to this assay. And now interestingly, on the left side you can see an illustration of the chip that will be used. Now in this case we will use a new chip since we have finished sequencing the previous one with the last sequencing run. And what you can see here is that now one of the lanes has been marked green, and that's in fact the lane that will be used to run these samples. So now what happens if I add a fifth sample? So let's choose a sample, assign this as well, and now I have 5 samples added. Now you can see that in fact 2 lanes have been marked green. This indicates that to run 5 samples for DNA and RNA we require 2 lanes. They do not fit on 1 lane. So let's remove the 5th sample again, or we could also fill it up for 8 samples to really make cost-efficient use of the 2nd lane. But in this case we can remove it, we click update, and now we're back to 1 lane. So now we have chosen our samples, and the last step is to provide the concentrations of these samples. This you can enter here for each sample, both the DNA concentration as well as the RNA concentration. You can see here a concentration range ranging from 0.67 nanogram to 2 nanogram per microliter. This actually can be defined by the user. The default value basically requires 10 nanogram of input DNA or RNA. Now what happens if we have more? We can load that, and then you can see here on the right, on the dilution factor, the instrument will actually automatically dilute to the highest value that is chosen here in our range. So this is optional. We can also decide we dilute all our samples manually, but we have the option to actually let the instrument dilute the sample to the right concentration. So we would type in our concentrations here, and maybe one thing, if we have less than the minimum, let's say we have 0.5 only, we get a small exclamation mark here telling us the sample concentration is out of the defined range. That doesn't mean that we cannot run this sample, it's just a warning saying, look, you're below minimum, there could be issues. In our experience, in fact, very often there is no issue and the sample still works. So for those of you who are familiar with other NGS assays, you may know that sample quantity is not the only thing that matters, it's actually even more so the quality. So even when we have very little, even below 10 nanogram, in our experience with the precision assay, very often we still get very good results. So once we have added our concentrations, we will press next, and what we will get is an overview of the reagents here on the very top. On the consumables required for this run are all the reagents listed that we now have to prepare. Some of them are frozen, some of them are in the fridge. The ones that are frozen we should now thaw around 30 to 45 minutes before we start the instrument. Then in case we have already done 1 or 2 lanes on the chip, the sequencing reagents can actually be reused, so they will already be installed on the instrument and will be listed here on the consumables, installed and ready to use. In this case we will start from a new chip, so these are not loaded yet. Then we have the number of primer tubes that we will require for the library preparation, depending on the number of samples that we have. And last but not least, again we get an overview of our samples along with the concentrations that we have added, and most importantly we get an overview of the scheme of how we need to pipette our samples onto a 96 well plate, both the DNA and RNA sample. That's in fact the only pipetting step of the entire process that we will have to do. So we would save this run, and now basically we are done with planning the run, and the next step is to go to the instrument and load it according to the loading scheme. So now I've shown you how to set up a run plan in the Genexus software. The next step is to go to the instrument and start the actual run. The first step to do this is to press the run button. Now the system will initialize. Next, we will choose the plan that we have created now in the software. Again, we get a quick overview of the assay as well as the number of lanes that will be run as part of this particular run. Again, here we can see that we have 2 times DNA and fusions on both lanes. When we confirm, we press next. So before we can load the deck, there will be a 2-minute UV clean to make sure that the instrument is sterile. And then we need to load the reagents. So here I have prepared all the reagents required for this run according to the run plan that I have created. First, we have our panel, the primers, required for target amplification during library preparation. Next, we have the barcodes. These are used to tag the individual libraries in order to distinguish them after sequencing. Then we have here strips 1 and strips 2 that are required for library preparation as well. Next, we have strip 3 and 4, and these are required for template preparation as well as sequencing. Of course, the instrument requires pipette tips as well as 2 empty 96 well plates. Then, very importantly, we have the sequencing chip along with the adapter that is required for the instrument to be able to load the libraries onto the chip. And last but not least, but very importantly, we have here our samples where we have pipetted our DNA and RNA samples according to the run plan that we have created. So now that all our reagents are ready, we can load them on the instrument. And in fact, if we look at the screen, it tells us step by step which reagents we have to load. And in fact, there are 3 cameras inside the instrument that are checking whether we are placing the right reagents at the right place. And if we do not, it will give us a warning and tell us that we need to fix something. So let's start with the primers. And I can illustrate to you also here what happens if we, for example, forget to take off the caps from the primer tubes. So I will place it in like this. And now you can see that the position where I placed the primers is turning orange, indicating that there is an issue with this position. So now I can remove the caps as it should be done and reinsert the tubes into the position. And as you can see on the screen, now it's showing up as gray, indicating that it is correctly inserted. So I will now continue to load the other 3 primer tubes. So now I can see on the screen that all 4 primer tubes are correctly loaded, and it's telling me to next load the barcode plate. And these go all the way in the back behind the primer tubes. Then the instrument indicates that I should load strips 1. Again, here I just quickly want to illustrate you what happens if I load the wrong strip. So if instead of strip 1 I load strip 2 in the strip 1 position, the screen turns orange, indicating that there is an issue with that specific position. So now if instead I load the correct strip 1, as before, you will see, I can see on the screen that it is now turning gray, indicating that it is correctly inserted. So now I will load the other strip 1 for a total of 3 strips. Once these 4 strip 1s have been correctly inserted, the system is now asking me to load strip 2. Next, strips 3 and strips 4. Next, we have to load the pipette tips. Then it is asking us to load the empty 96 well plates into the thermal cycler and the other one here into the magnetic rack that is used for enrichment after template preparation. Then finally we get to add our samples that we have pipetted onto the 96 well plate covered with an aluminum foil. And last but not least, the chip along with the chip adapter. And of course also the adapter that will be used to pipette the samples onto the chip. And now that I have loaded all the reagents, the instrument is asking me to close the latches that hold the strips in place. And finally, it asks me to close the door. So once we have loaded everything on the deck and the door is closed, the bottom doors are opening, allowing us to provide the sequencing reagents. Here we need to provide running buffer for the sequencing reaction. We have a cleaning bottle, as well as an empty bottle for waste. And last but not least, we have here a cartridge containing nucleotides that will be required for the sequencing reaction. So I will start with loading the running buffer bottles. Next, the waste bottle and the bottle with the cleaning solution. Then, last but not least, the nucleotides. Once all the reagents for sequencing have been loaded into the bottom of the instrument, it is asking me to close the doors. And then the run will start. So now the instrument will take another 16 hours to complete the run until we have our final results. Previously, doing this manually in the lab would have taken us at least 2 to 3 days, and now we can basically get NGS results overnight. And I will be happy to discuss the results with you in 16 hours. So now I've shown you how to register a sample, how to set up a run, and even how to load the instrument. Now the last part I want to show you is how to look at the results once the sequencing run is done. So again, being back here, we now go to the results tab, where we have the option to look at the results on a sample by sample level or on the run level. So let's go look at specific samples. So we can click either on the sample directly, or we can in fact go to the overall run. And this is what we do in our daily routine. So we first look at the overall run performance. So when we click here, we now get an overview. On the right here, you can see an illustration of the lane that was run. In this case, this was actually a lane 3 of a chip where this sample was run on. On the left, you can see some metrics, overall loading of this lane, the enrichment, so how many of the loaded wells on the chip actually had DNA on them, and some other QC metrics. So once overall QC is done, we go sample by sample. And now I would like to focus on one sample, which is here, this E2747. If I click on the sample now, I will be brought to a key findings landing page. And this is a page where I can again see a summary of the sample details. I can again see which was the cancer type, the percentage of tumor cells, and I also get some QC metrics for this sample, many of which were already visible on the previous page, like average base coverage depth, uniformity of base coverage, percent on target, and also median molecular coverage, so how many individual molecules for each region did we detect. And then most importantly, of course, we immediately get a view of the key variants that were detected. In this case, non-small cell lung cancer sample, and we detect an EGFR exon 19 deletion. We get some additional information here for the allelic frequency, which in this case is 18.6, which more or less fits the estimated tumor cell content. Also visible on this site is actually the coverage of key genes, tumor-specific key genes that we can define ourselves. In this case, for non-small cell lung cancer, we have EGFR, we have BRAF, HER2, KRAS, MET, and then also in case we run the RNA assay with the DNA, we also have immediately visible whether we have detected a fusion in ALK, ROS1, NTRK1, 2, 3, or RET. Below we get some additional information on the coverage of these key genes, so that we can immediately see if we don't detect a mutation in a specific gene, we can immediately see whether the coverage of that particular gene was actually good, as you can see here. So this is available for all the genes, where you can then see here even the hotspots that are indicated. In orange are the amplicons, and in green is the exon structure. And here we have an exon 19 deletion, so you can see this here. this is indicated here as well, the detected mutation. So that's just an overview of the key findings. Then we can go into more detail, so we can look at the variant in detail by clicking on the Variants tab. And now here again we have it. The type is a deletion in EGFR gene. This is the amino acid change, so very common exon 19 deletion. And when we click on the variant, we now see in the bottom an IGV window that opens. And now we can configure this. We usually go into squish mode, and now we can already spot here the deletions. I sort by base, and now I have all my reads containing the deletion nicely sorted, and it gives me just a visual confirmation that the called exon 19 deletion is indeed present. Alright, so then of course we can get additional details on the variant. We can see the precise nucleotide change back here as well, and again we can confirm this by looking at the IGV, the integrated IGV. Once we're happy and confident about the call, we can actually download the files, the required files that we want to store. We can download the BAM files, we can download a summary of the detected variants, and this is what we do. We download this, we archive it, and from this we then actually generate the string that we will then import in our integrated pathology report. So this is done via download files, and I can choose here really what I want. I can download the SNVs, the CNVs, the BAM files. Importantly, of course, also the QC metrics. So we do this as well just to archive, to have everything in one place in case we go back sometime later to that case and we have everything in one place. So the next sample I want to show you is again a non-small cell lung cancer sample. But this time it's an RNA-based fusion detection analysis that we have performed. So let's open the sample. Again, when I click on the results, I immediately get thrown to the key findings page, where I can see again some sample details, and in fact I can immediately spot that there was a RET fusion detected. And now in fact this is detected on two different levels. Once it's detected as a targeted fusion partnering with KIF5B, and then it's also detected as an exon tiling imbalance, which is the part of the assay that allows us to detect novel fusions, so fusions with unknown partners that are not specifically targeted. So now in this sample we have in fact two lines of evidence for a RET fusion. Of course, one, we have the targeted assay that detects K5B RET fusion, and we have the additional evidence of the exon imbalance. Again, here we will download the data and put it into our integrated pathology report. So the next sample I would like to show you is a colorectal cancer sample. So again, let's go straight to the sample results by clicking on the sample name. Let's go to the key variants that were detected. In this case, it was BRAF V600E that was detected at 40.6% allelic frequency, along with a GNAS R201C mutation at the same allelic frequency. Again, we can go into the variant details. We can look at the variant in more detail in IGV by clicking on the variant here. And then in this sample, we would report both the BRAF and the GNAS mutation and put it in our report. All right, the next sample is from a gastrointestinal stromal tumor, where we check mainly for mutations in KIT as well as PDGF receptor alpha. So let's jump straight in. Let's have a look at the QC first. Key variants detected here, we can see a mutation, V560D mutation in KIT at an allelic frequency of 47.2%. Looking at the tumor cell content of 90%, again, this fits very well. And looking at the detail, just to verify it, and we will click here again, we will see the reads in IGV,